Welcome back, Biology Dominators! Day 29. We are in the final stretch of this crash course, and today we're covering one of the highest weightage, most application-based chapters in NEET Biology, Biotechnology, Principles and Applications. This chapter is pure marks if you understand the logic, so lock in, no distractions. Let's go! First, what is biotechnology? The NCERT defines modern biotechnology as techniques using living organisms or their derivatives to make products or processes for specific use. But NEET loves to test the three pillars of modern biotechnology. 1. Genetic engineering to alter DNA. 2. Maintaining sterile or aseptic conditions, and 3, bioprocess engineering to scale up these processes. Remember this trinity. It has come in PYQs multiple times. Now the tools. This is where most NEET questions are hiding. Restriction endonucleases. They are the molecular scissors. They recognize specific palindromic sequences on DNA and cut it. EcoR1 cuts at GAATTC, producing sticky ends. The term palindrome here means the sequence reads the same on both strands in the 5' to 3' direction. This is a very common MCQ trap. Then we have DNA ligase, the molecular glue that joins these ends together. And finally, vectors, the vehicles that carry foreign DNA into the host. Plasmids are the most common vectors in NEET. They carry selectable markers like antibiotic resistance genes. Here is the core process, rDNA technology. Step 1, isolate the gene of interest. Step 2, cut both the gene and the vector using the same restriction enzyme. This is critical because compatible sticky ends must be formed. Step 3, ligate them with DNA ligase, forming recombinant DNA. Step 4, introduce this into a host cell, usually E. coli, through transformation. Step 5, select the recombinants. This is where selectable markers come in. If the gene was inserted into the ampicillin resistance gene, that bacteria will become ampicillin sensitive. This is called insertional inactivation. Step 6, clone and express the gene. Every single step is NEET testable. PCR, Polymerase Chain Reaction. This is like a photocopy machine for DNA. 3 steps in each cycle. First, denaturation at around 94 degrees Celsius. The double-stranded DNA separates. Second, annealing. Primers attached to the single-stranded template. Third, extension at 72 degrees. Taq polymerase synthesizes the new strand. Taq polymerase is thermostable. It comes from the bacterium Thermus aquaticus. After the PCR, we use gel electrophoresis to visualize the DNA. Smaller fragments migrate faster through the gel. They travel further. The gel is stained with ethidium bromide and viewed under UV light. Both of these are very frequently tested in NEET. Applications in medicine. This is where NEET loves to ask questions. Number 1, insulin production. Before genetic engineering, insulin was extracted from pig and cattle pancreas. Now, the human insulin gene is inserted into E. coli using rDNA technology. The product is called Humulin. It was the first recombinant DNA pharmaceutical approved for human use. Number 2, gene therapy. The first clinical gene therapy trial was for ADA deficiency, which causes a condition called SCID, severe combined immunodeficiency. The ADA gene is introduced using retroviral vectors into the patient's lymphocytes. Number 3, molecular diagnostics. PCR can detect pathogens even at very early stages before symptoms appear. ELISA detects antigens and antibodies. Both are important diagnostic tools. NEET has asked about this multiple times.
Agriculture applications, extremely important for NEET 2026. Beet crops. The gene from Bacillus thuringiensis, called the cry gene, encodes a protein called cry protein or crystal protein. This protein is actually produced as a protoxin inside the bacterium and becomes toxic only when activated in the alkaline pH of the insect gut. Different cry genes target different insects. Cry1AC and Cry2AB target cotton bollworms, while Cry1AB targets corn borer. Golden rice. This was engineered to produce beta-carotene in the endosperm of rice grains, addressing vitamin A deficiency. And RNAi, RNA interference, is used in tobacco plants to introduce genes that silence specific nematode genes, protecting the roots. This concept of RNAi is new syllabus and a definite NEET 2026 hot topic. And that is day 29 complete. Biotechnology is a chapter where every concept directly maps to an MCQ. If you understand the logic, why the same restriction enzyme is used, why Taq polymerase is thermostable, why Bt toxin is a protoxin, you won't just remember the answer, you'll derive it. Tomorrow is day 30, ecology, the final chapter of this crash course. You have come so far. Do not stop now. Drop your doubts in the comments. We read every single one. Like this video, share it with your NEET squad, and I will see you tomorrow for the grand finale.